Good morning, good afternoon, and good evening. Welcome back to Chapter 10. We're talking about the process of transcription and translation. We're talking about the process of taking a DNA gene, following the directions, and making the product that it is the directions for. We've made it as far as making a copy. Our molecule called RNA polymerase unzipped their DNA and made a copy of one side of that gene. Well, now my piece of RNA is going to leave the nucleus. It's not going to leave just yet. We've got to finish it first. Enzymes within the nucleus will finish my piece of raw messenger RNA by cutting out bits and pieces that aren't supposed to be there. Your gene, that recipe is actually a lot more complicated than simply just a cup of flour, a cup of sugar, and 2 eggs. It's a lot more complicated than that. It's basically, if you could imagine a recipe for a cake. And after you put together the base pieces of it, it says, all right, now add a cup of chocolate chips and a cup of peanut butter chips and a cup of walnuts and a cup of macadamia nuts and a cup of white chocolate chips. And you're like, this is way too busy. But what this one gene actually is the recipe for a half a dozen different cakes. You cut out the extra bits and just leave in the cup of chocolate chips. I cut out all the extra pieces and say, no, you don't need to add all those bits. And then you glue the recipe back together again and say, all right, this is how you make a chocolate chip cake. That same recipe, if I leave in the macadamia nuts and take out all of the rest of the extra cups of stuff, I glue that back together again, and now I've got a macadamia nut cake. All right, so one recipe is actually the recipe for several different proteins that are variations of each other. Well, you have to make sure that you put together just the right bits of it. And this is what the enzymes do before the messenger RNA leaves. They cut out the extra pieces and glue the proper pieces back together again. The extra pieces that get removed are called introns. The pieces that get glued back together again are called exons. And it's a little bit tricky. It's like, that doesn't make sense. Well, just remember that the introns, the pieces that get cut out, stay in the nucleus. And the exons, the bits that get glued back together again, get to exit the nucleus. The exons are the finished product. The piece of messenger RNA is glued back together again, and it's just the recipe for the chocolate chip cake, not chocolate chips and walnuts and peanut butter chips and white chocolate chips and not all that other junk, just the bits and pieces to make this one recipe. Now that I have my finished, we call it mature, messenger RNA. Now that I got my piece of finished RNA, messenger RNA, it leaves the nucleus. But now it's time to follow the directions. Now it's time to read the code on the RNA and follow the code to put together the amino acids in the right order. And let's back up for just a second, back all the way up to chapter, 2 or 3. And let's talk about, remember, the three levels of protein structure. The very first level of protein structure is that amino acid sequence. You have to put the amino acids together in the right order so it twists and folds in the right places, so it twists and folds in the right places to get that very specific three-dimensional, that tertiary functional conformation. The way you have to get to that third state, that third level, is by following the first level. I've got to make sure the amino acids are put together in the right order. The messenger RNA, that's what the code is for. It's to tell you which amino acid to put first, which one to put second, third, fourth, fifth. You can think of a protein very much like a word. I have to put the letters together in the right order to spell the right word, and if I don't put the letters together, if I mistake one letter for another, I've changed the word. And a lot of times, if you change the word, you broke the word. It doesn't work. It's misspelled to the point where you can't understand what you're being told. Proteins are just like that. They're really specific. I can't put the wrong amino acid in a place, in a spot, because if I do, I will break the entire protein because I put one in the wrong spot. The messenger RNA leaves the nucleus and it encounters a small subunit of a ribosome. Remember, ribosome is made of 2 pieces, a small subunit and a big subunit. It encounters a small subunit first. Once the small subunit has stuck to the piece of messenger RNA, now the big subunit, the large subunit, attaches to it. and it literally pinches the messenger RNA between the two halves of the ribosome. What is that code? Remember that DNA is A's and T's and G's and C's. Also remember we've taken the DNA code and we've translated it, transcribed it, into RNA. So now we're not talking about A's and T's and G's and C's, because now we've made an RNA copy. Now we're talking about A's and U's and G's and C's. There are 4 different bases. A, U, C, and G. That's it. Those are my 4 bases. I've got to ask for 20 different amino acids, because that's how many there are. There are 20 different amino acids that are used to build all the proteins on the planet. There are 20 different ones. So somehow these 4 letters have to be a code for asking for 20 different amino acids. Well, if A asked for 1 amino acid and U asked for another one, that would only be 4, right? A asks for 1, U asks for 1, C asks for a different one, G asks for a different one. I can only ask for 4. Well, 4 isn't enough. I've got 20. So I have to have more codes than that. You have to use 3 of these in a row. 3 bases in a row is enough to give me enough codes to ask for 20 different amino acids. The problem is there are 64 combinations of 3 bases in a row. If I've got 64 codes for 20 things, how many codes per thing are there? Well, 64 divided by 20, 6 divided by 2, there's about 3. There are about 3 different codes that ask for any one amino acid on average. But to be fair, to be honest, I don't think there's any one of them that's actually 3. It's usually like 2 or 4 or 6. 2 codes for this one, 4 codes for that one, 6 codes for this other one. That's how the codes work. 3 in a row, 3 bases in a row on messenger RNA is called a codon. AUG is the start codon. We said that the piece of RNA polymerase has to start on that starting line. Well, every single gene begins with the same code. So every single piece of messenger RNA begins with the same code, and that same code is AUG. AUG asks for one particular amino acid, and the one particular amino acid asks for is methionine. The other 63 codes, 3 of them are the stop codon. We said that when the RNA reaches the end of the line, it falls off. When it reaches the finish line, it comes off of the DNA. Well, the codes on messenger RNA that are the finish line is any one of these. It doesn't have to be all of them. Any one of them work. So UAG, UGA, UAA, those are the stop codons. That means get off, you're done. Okay? So if there's 64 total codes, 64 combinations of 3 in a row, well, here's 4 of them. So there are about 60 codes left. This really big, complicated chart shows all 64 codes. No, you're not responsible for all 64 codes. The only codes you need to know are those 4 that was on the previous slide. The start code, the start codon, and the 3 stops. But as you can see, most of them have multiple codes. Like here, phenylalanine, it's got 2 different codes. Serine, it's got 4 codes here. Oh, serine also has 2 more codes over there. So here's one of those. It's got 1, 2, 3, 4, 5, 6 codes. Leucine, 1, 2, 3, 4, 5, 6 codes. Most of them have like between 2 and 4. Like 4 here, 2, 2. Most of them have between 2 and 4 codes. Methionine, I think, just has the 1, the start codon. Where methionine went. I can't see it at the top of my head. There it is right there. Methionine just has the 1. But anyway, these are all of the 20 different amino acids. So we said that a gene is like a recipe. I make a copy of that recipe, and I take it to the kitchen to understand what the codes mean. Instead of seeing something weird like methionine or isoleucine or phenylalanine or whatever, imagine that the cook is standing at the counter and he's reading the recipe. And the recipe says that I need a cup of chopped carrots and I need half a cup of diced onions and I need a half a cup of celery. What the chef does is he doesn't say, bring me a cup of carrots. He just calls for Sarah. Sarah. All Sarah does is chop carrots. And when he calls Sarah's name, she brings what she always carries. And what she always carries is a cup of carrots. Now, it just so happens that Jim and Taylor and Connor also only chop carrots. And all he has to do is call one of those four names. He just calls one of those four names, whether it's Sarah or Jim or Connor or Taylor. It doesn't matter. If he calls any one of them, they'll all bring him the same thing, which is a cup of carrots. If he calls for Jennifer always brings a half a cup of diced onions. But it's not just Jennifer. It's also Amanda. And it's also Sherry and Jeff and Matthew and Clark. Any one of those people, he just has to call their name. Any one of these. And if he calls any one of those people, they always bring the same thing, which is half a cup of chopped onions. That's how the code works. Each combination of three asks for one particular ingredient. The ingredient happens to be an amino acid here, but that's what the codes mean. That's how it works.